Over the years of 1950s, there has been dispute about if DNA replication is semi-conservative, conservative, or dispersive. Although it seems to many people that a semi-conservative is more reasonable, since we sort of know the mechanism of replication. Yet scientists won't say a word without proving it in experiments beforehand. Now, before we look into the story of the research by two American scientists, let's break the big word down. What on earth is semi-conservative? Well, literally, semi means half, and conservative means keeping the old stuff. And here, semi-conservative just means that a product of DNA replication will not comprise of two new strands. Instead, it will contain a new strand with an old one bonded together by hydrogen bonding. James Watson and Francis Crick was awarded Nobel Prize for their groundbreaking research on DNA structure, which outlined the basic structure of DNA we see today. Based on the preceding work done by Rosalind Franklin and other relevant work, they managed to hypothesize that two strands of a DNA molecule separate during replication, and each act as a template for a new strand. This hypothesis is a well-known semi-conservative hypothesis. It wasn't until 1958 when a hypothesis was proved in a scientific experiment conducted by Meselson and Franklin-Stahl, namely the Meselson-Stahl experiment. In order to test the hypothesis, they intended a method that separates macromolecules according to the difference in their buoyancies, along with an approach called equilibrium density gradient centrifugation. They carried out the experiment with isotopes of nitrogen, nitrogen-14 and nitrogen-15. Nitrogen is a major constituent of DNA molecule, and nitrogen-14 exceeds the number of nitrogen-15, which is heavier but not radioactive in the molecules. FYI, isotopes are just atoms of the element that have the same proton number but different neutron number. So the nitrogen-15 is heavier because it has one more neutron. In the experiment, they prepared food source that contained ammonium chloride as a source of nitrogen, and then grew populations of E. coli, a type of bacteria that could be found in the alimentary canal. The nitrogen in the ammonium chloride was nitrogen-15. They left the bacteria long enough so that nearly all the bacteria only have nitrogen-15 in their DNA molecules. They subsequently divided the bacteria into portions. The first portion was transferred to a food source that only nitrogen-14 presents, and the second portion was left for about 15 minutes, permitting DNA replication only to occur once. And the last portion was left long enough to allow DNA to replicate 2 or more times. DNA was extracted from the bacteria from 3 groups and added into sodium chloride solution, after which is placed into a centrifuge. The heavier the molecules are, the closer they rested near the bottom. In the first test tube, in which bacteria were grown with only nitrogen-15, band of DNA is found near the bottom of the solution. In the second test tube, the band is found less near the bottom of the central part of the solution. The bacteria inside was one generation of replication with nitrogen-14. In the third test tube, band of DNA was extracted from the second generation with nitrogen-14. 2 bands can be found with nearly equally length in the central part. In the last test tube, was extracted DNA from the third generation of bacteria with nitrogen-14. Two bands are also found with a longer upper band and shorter lower band. So the question is, when exactly is DNA replicated? The cell cycle consists of M phase, mitotic phase, when cell division occurs, and I phase, anaphase. DNA replication happens during interphase, more specifically S phase of anaphase, that is, synthesis phase. Anaphase is essentially the daily life cycle of the cell. Cells spend most of their life in anaphase, and cell division occurs during M phase. Now, everyone makes mistakes. However, sometimes mistakes can lead to severe consequences. When it comes to DNA replication, single mistake can result in huge flaw in cell functionality. Now, how do you ensure mistakes are not made during DNA replication? Well, as we've already known, DNAs are molecular polymers of nucleotides. DNA polymerase, which assemble nucleotides to original strain, only link complementary nucleotide to the growing new chain. This ensures mistakes are barely made. The possibility of mistake is 1 in every 10 to the 8th pairs. In addition to avoid mistakes, DNA polymerase also take approaches to check for mistakes and fix them. The process in which they check for mistakes is called proofreading. During proofreading, when they detect a nucleotide is mismatched, they will immediately stop and use what's called 3 to 5 exonuclease activity to remove incorrect nucleotide. For those mistakes not fixed by proofreading, mismatch repair is used to correct further errors after new DNA has been made. During its process, a protein complementary recognizes and binds to the misfire base. A second complex cuts the DNA near the mismatch. More enzymes then cut out the incorrect nucleotide in a surround patch of DNA. A DNA polymerase then replaces the missing section with the correct nucleotides, and DNA ligase seals the gap in the DNA backbone. Body also has various mechanisms after DNA being damaged, including chemical reversal, excision repair, and double-stranded break repair. Due to the time constraint, it is a story for another time. Thanks for watching.